If we see the internal structure of kidney, there we can identify some structures. A typical kidney of an adult, there is some measurements in that particular person's. So here we can identify some measurements like length, width and thickness. So if you see a typical kidney, there we can identify about 10 to 12 centimeters of length and 5 to 7 centimeters width, and along with the 2 to 3 centimeters is thickness. So here we can say that from the anterior end to the posterior end, we can identify measurements that is about 10 to 12 centimeters of length. And if you see the width from this area to this area, so that is about 5 to 7 centimeters in width. If you see the thickness is about 2 to 3 centimeters is the thickness. And if you see that the longitudinal section of kidney, we can identify two parts. So here we can identify the two parts are there. They are here, that is the reddish colored structure, and this is outer surface of the kidney, and whereas the inner surface is there, that is called as another part. So that is the outermost part of the kidney is known as renal cortex, and the innermost surface of the kidney is known as medulla. So here we will see that the longitudinal section of kidney shows two specific parts, outer cortex and the inner medulla. So here we can identify the cortex, and inside of that medulla is identified. And next, here the renal cortex. What is renal cortex here? The renal cortex is the superficial light red area of kidney. This is called as light red area of kidney. That part is known as cortex. So cortex is the outermost surface of the body part of kidney, and that is appears like superficial light red area of the kidney. And next, renal medulla. So renal medulla, I mentioned that innermost part of the kidney is known as renal medulla. And here, deep dark reddish brown area of kidney. So how can we say that? How can we identify that medulla part here? It appears in the deep dark reddish brown area of kidney, that is considered the renal medulla. And next, it is divided into multiple cone-shaped structures. So here we can identify multiple cone-shaped structures are there. They are called as renal pyramids. And here the base of each renal pyramid, this is the base of each renal pyramid, that faces towards the cortex region. This is called as cortex. The base of each renal pyramid faces towards the cortex region, and the apex of each renal pyramid is called renal papillae. So here we can identify the renal papillae, the base of or apex region of the each renal pyramid included with the renal papillae. And next, the renal papillae project into the cup-like structure called calyces. So here the calyces are there. Here cup-like structures will be present. They are called as renal calyces, and the renal calyces are formed by the renal papillae, and they are projected into the cup-like structure. They are called as renal calyx or renal calyces. Next, the portion of renal cortex extends between the renal pyramids. They are called as renal columns or columns of Bertini. So here you say that the renal cortex extends. This is called as renal cortex extends between the renal pyramids. This is the renal pyramid, and there we can identify the renal columns or columns of Bertini. So that structures are identified in the internal structure of kidney. And next, within the renal cortex and medulla of each kidney contains 1 million of nephron. If you see that the total one kidney, if we have taken, then we can identify 1 million of nephrons will be identified. Here within the renal cortex, this is called as renal cortex. Within that, nearly 1 million of nephrons will be identified. So that is each kidney included with 1 million of nephrons. Here we can say that both the kidneys having the 2 million of nephrons. And next, calyces. What are calyces here? So calyces are the cup-like projections. They are formed from the renal papillae. Here we mentioned that renal papillae projections. The renal projections, renal papillae projections forms like cup-like structures. They are called as calyces. Here there are two types. Minor calyces are there, at the same time major calyces are there. Here minor calyces, they are called as funnel-shaped structures and open into the renal papillae. So these are the renal papillae. So there we can identify the here minor calyces. These are called as minor calyces. They are located in the renal papillae and they are open funnel shaped structure called renal papillae. So they are in the form of 8 to 18 minor calyces are present in each renal papillae. And next, major calyces. They are formed by the merging of minor calyces. Minor calyces are merged each other and forms the bunch of characters called major calyces. They are of about 2 to 3 in number and they converge to form the renal pelvis. So all are united to form and a sac-like or tube-like, large tube-like structure is formed that is called as pelvis. So pelvis opens into the ureter and whenever the filtration is finished, the remaining parts of nitrogenous waste material passes into the pelvis region, from there enters into the ureter. So these are the structures we can identify in the internal structure of kidney.